today's Tech Talk on managing manufacturing changes for the radiation process. I'm Sarah Conway, Lab Technical Manager with STERIS Applied Sterilization Technologies, based out of our Tullamore site.
And I'm Jason Rogers, Principal Scientist out of our Brooklyn Park Laboratory.
In today's Tech Talk, we will review the microbiological laboratory testing for validation of radiation sterilization, looking at the applicable standards, the method validation, and the testing process itself. We will also look at the maintenance of the radiation validation by dose audit studies. And then we will look at managing manufacturing changes. Under this topic, we will examine different types of changes that we may encounter in manufacturing operations and look at things to consider around these changes. The standard references for guidance on the radiation sterilization validation and associated microbiological tests are listed here. ISO 11137-2. In this standard, it guides us on the processes used to validate Method 1, Method 2, VD-Max 15, and VD-Max 25. ISO 13004 outlines additional VD-Max dose options from 17.5 kilogray up to 35 kilogray. From the microbiological testing point of view, we look at the standard 111737-1. In this standard, it covers bioburden testing, including the testing of inhibitory substances and bioburden recovery efficiency. It also gives guidance for product families for bioburden testing and data use and trending. In ISO 11737-2, the standard provides information on sterility in testing, including the sterility method to suitability, which is B&F testing, and the validation of the sterility test method. The microbial testing of a radiation sterilization validation occurs as 4 phases or stages. Phase 1 is the test method validations. This ensures accurate microbiological test methods for the device, i.e. giving us no false negatives and making sure we have acceptable recoveries. Phase 2 is the enumeration and characterization of bioburden. Here we're determining the number and the type of bacteria or fungi on a medical device prior to exposure to the radiation. In Phase 3, the determination and calculation of verification dose, we're using the bioburden data and the applicable table in the ISO standard to determine the verification dose. And finally, in Phase 4, the verification dose experiment, here we're performing the irradiation of the product to the verified dose range and then performing the test of sterility. Regarding Phase 1, test method validations, these are a very important part of the radiation sterilization validation, as they ensure that the results we find are accurate and appropriate. There are 2 considerations for test method validations used in the radiation validation process: bioburden recovery efficiency testing and screening for inhibitory substances. This is both bioburden test related and sterility test related. Test method validations are required by the standards and expected during device review and audits by regulatory bodies. For the bioburden test method validation, it is a critical component of the routine bioburden process, as it ensures that the test method to be employed will give true data, essentially ensuring that acceptable recoveries are obtained and there isn't a gross underestimation of the product bioburden. Organisms may be more or less recoverable from different materials or substrates, and this test measures and corrects for the recovery of the test samples. Bioburden recovery efficiency testing validates the removal and culturing of organisms from the product and ensures accurate measurement of contamination on or in a sample. There are two methods that we can look under this process: native repetitive method or inoculation method, which is direct inoculation. Screening for inhibitory substances is important to ensure that the product does not reduce or affect the detection of microorganisms in our test. This test validates the neutralization of any substance that may be inhibitory or lethal to microbes from the testing technique employed for routine testing. Screening for inhibitory substances ensures that we are able to detect and measure contamination on a sample without the concern of falsely low or falsely negative results. Often filtration, dilution, or inactivation removal of the inhibitory substances are methods for mitigating the substances. There are two inhibitory substance screening tests used in the radiation validation process. The first of these is Bioburden inhibitory substance screening. Bioburden test, which is the Bioburden test method suitability. This test ensures that product present in the extraction fluid does not give a reduced bioburden count. It is a quantitative test resulting in a number of CFUs obtained and compared to control samples. The required outcome is the growth recovered from the samples containing product is not significantly different to that seen in samples containing no product, i.e. the positive controls. The completion of inhibitory substance testing may not always be applicable, but the consideration should be done and documented by the manufacturer and maintained in the quality system. The second inhibitory substance screening test is the bacteriostatus and fungus status test, or BNF for short. This is the sterility test method suitability. This test ensures that the product present in growth media does not create a falsely negative or no growth result. This is a quantitative test, so presence or absence of growth, and is tested against several compendial organism strains. The required outcome for this test is that there's growth of the test organism in the presence of the product within the incubation period. Now that we have validated test methods, we can perform bioburden testing to determine the number and type of microorganisms present on the product. Bioburden is one of the most critical components of the radiation validation process. The number of microbes from this test is used to determine the verification dose which will be applied to the product, and the type of microbes affect the potential resistance to radiation. Understanding the number and type of microbes are also important factors in the grouping of products into product families, the inclusion of product into an existing family, or the selection of product to represent a product family. Bioburden testing is performed in accordance to guidance from ISO 11737-1, and the verification dose is most often determined from tables within ISO 11137-2 or ISO 13004. Again, this is a quantitative test, so we have a specified number of organisms as a result of which is reported as CFUs or colony forming units. For a full radiation sterilization validation process, 10 units from each of 3 production lots are tested with the validated bioburden method. The bioburden determination should be carried out on product that have undergone the manufacturing process through final packaging. The product must also be reflective of routine device manufacture and the contamination that may be present for the routine sterilization process. Once cultured and enumerated, we can characterize the type of organisms present on the product. Characterization of the CFUs present on the device give insight into the likely sources of bioburden and their potential resistance to sterilization. The identification of the organisms can also assist in understanding how to appropriately group products into family or with the selection of a master product. Microbial characterization is also useful during investigations on exceeded bioburden limits or sterility failures. Most basic forms of identification are selective culturing, colony morphology, and gram staining. Selective culture uses select medias, heat shocking of extract, or incubation conditions to detect certain types of microorganisms. Colony morphology gives a visual description of the colony's growth characteristics, such as color, shape, consistency. The visual description helps to classify observed phenotypes over time when coupled with further identification processes, such as gram stain, and it can give insight into the possible genus or species recovered. To obtain a higher confidence level match, genetic identification or MALDI-TOF identification testing can also be completed. MALDI-TOF, or matrix assisted laser desorption ionization time of flight, uses ribosomal proteins analyzed by mass spectrometry to compare spectra to a library of spectra from known organisms. In a full radiation validation consisting of 3 independent patches, the verification dose is set using the overall average bioburden. However, if any single batch of the 3 tested has an average of twice or more the overall average bioburden, then the single batch average, i.e. the highest batch average bioburden, is used for dose setting. We can look up the bioburden average in the corresponding table in 1137-2 for the corresponding doses. So table 5 for method 1, for example, and table 9 for VDMAX 25. There are additional tables in this standard are also available for other VDMAXs. And then in 13004, we can look for doses for VDMAXs from 17.5 right up to 35 kilogram. Once the verification dose range is determined from the bioburden results, samples are then irradiated at the range from the verification dose experiment. For method 1 and method 2, 100 samples are required for the verification dose study. For VDMAX method, 10 samples are required for the verification dose study. Dosimetry used to measure the dose provided to the samples. Verification dose range determined has a 10% plus or minus range. If the dose used is less than 90% and the result of the sterility tests are negative, the results are acceptable. Test of sterility is performed for these samples and results are evaluated in accordance to the corresponding radiation standard. Once there is a validated radiation sterilization dose, dose audits are performed on a regular basis to ensure maintenance of the validation. ISO 11137-2, section 10.1, dose audits are carried out to confirm the continued appropriateness of the sterilization dose. This dose audit confirms that bioburden is under control and that the sterilization dose continues to achieve the required sterility assurance level. Dose audits are not required during periods in which the product is not produced. When infrequent production is performed, dose audits with each production batch may be appropriate. The frequency for carrying out dose audits is outlined in ISO 11137-1, section 12.1. Section 12.1.3.1 indicates that intervals of time between dose audits can be either 3 months or a different time interval can be selected based on documented rationales. In section 12.1.3.3, it is outlined that 12 months should be the max interval between dose audits. ISO 11137-1, section 12.1 also indicates the following around dose audit frequency. It outlines what needs to be considered if you want to increase the time interval between dose audits, what needs to be done with dose audits when product manufacture is infrequent, and what to do with regards to frequency of dose audit studies if a sterilization dose audit is unsuccessful. So what needs to be completed for a dose audit study? 10 samples are tested for bioburden regardless of the validation type, so be that VD-max, method 1 or method 2. The following exposure to verification dose, the samples are then tested for sterility. Depending on the validation type, different numbers of samples are required for this sterility test. So for VD-max, 10 samples are tested for sterility, whereas for method 1 or method 2, 100 samples would be tested.
After reviewing the validation and dose audit process, we can now begin to review some potential change scenarios and how they might be assessed. It is important to note that this presentation is not intended to be an exhaustive list of all potential changes or situations. There may be specific device or process-related changes that are not covered in this webinar, or may be best handled differently given specific circumstances or interpretation or acceptance of risk. The manufacturer is responsible for evaluating the impacts of changes to their medical device products and manufacturing process. This presentation may mention topics such as dosimetry, product functionality, biocompatibility, and packaging studies. The primary considerations are from a microbiologist perspective and the radiation process outlined in previous slides. As ISO 13485 details in section 7.3.9, with any change in the quality system, there is a need to document planned changes through a formal change control process and determine the significance of the change to function, performance, usability, safety, and applicable regulatory requirements for the medical device and its use. Records of changes and their review and any necessary actions are maintained. So how should manufacturing be documented? Here we have an example of a change control process flow, which is likely similar to your organization's change control process. Starting with a proposal or requested change, there is an analysis of the impact. Required stakeholders must approve or deny the request for change. If approved, planning and potential testing of the proposed change is performed prior to implementation. Once the change is implemented, there may be a review of effectiveness and a communication of the change before the change control is approved for closure. The ability of manufacturers to understand the impact of the change, verify through testing, and monitor effectiveness of a change is crucial to patient safety. As part of this change control process, the following items may be included: an assessment of the impact of the change to any regulatory filings. There may also be a review or comparison of technical documentation, suitability studies, and/or equivalency studies performed as part of planning or impact assessment of the change. Often an area that is used or useful in historical context is documentation of decisions with any rationale or justification. Review of any process risks, risk assessments, such as process FMEAs, that might be impacted by the proposed change should also be documented and updated if necessary. Note, all associated records for these activities should be maintained in the manufacturer's quality system. Product bioburden is used to determine the verification dose that will be applied during the verification dose experiment, and which substantiates the routine sterilization dose, as product bioburden, i.e. the number and types of microorganisms present on a product, is a critical component of the radiation validation process. Any changes that impact it should need to be understood and formally assessed. Under radiation validation process, product bioburden is also a key consideration in determining product families and products to represent a family. As outlined in ISO 11137-2, Section 4.1, definition of product families is based principally on the number and types of microorganisms present on or in the product, the bioburden. It also goes on to discuss that products within the family have bioburden with similar numbers and types of microorganisms. Anytime there is a change in quantity of microorganisms or change in the type or resistance of microorganisms, the impact to the product family definition or the selected representative device needs to be assessed or considered. ISO 11137-2, Section 4.4.2 also states modifications to product and/or modifications to the manufacturing process shall be assessed through a formal documented change control system. The continued effectiveness of the established sterilization dose can be demonstrated by carrying out dose audit studies. Dose audits may be used as part of the change control process to confirm the change has not impacted the validated sterilization dose. As bioburden and sterility testing are performed under dose audit study, additional useful information on product microorganisms can also be maintained or obtained by performing identifications on bioburden plates or any positives obtained during sterility testing. The frequency at which dose audits are performed may need to be considered or adjusted when changes to the product or manufacturing process are made, which impact product bioburden, the quantities and types of microorganisms present on the product. For instance, if a longer frequency between dose audits is justified beyond quarterly, the change may impact the justification or may require more data to ensure that the control of bioburden is maintained. ISO 11137-4, Section 8.2 provides guidance to assess changes and their impact to dose mapping studies. For instance, changes to product orientation, and packaging should be assessed for impact to existing dose mapping studies and could vary depending on the technology used. While LAL is not part of the radiation validation process itself, it may be a test that is performed routinely as part of batch release when product is irradiated. If this is the case, changes to the product or manufacturing process should be assessed with regards to the impact on endotoxin levels and the suitability of the test method. As per the new standard on endotoxin, ISO 11737-3, Section 9.2.1, changes to either the product or manufacturing process or both shall be reviewed to determine whether they are likely to alter bacterial endotoxin levels with consideration to the purpose for which the bacterial endotoxin testing data are to be used. For any changes that have noted a potential impact on bioburden quantity and/or type, considered with regards to the impact on the endotoxin levels should also be examined. It is important to note that the impact of a change to endotoxin levels cannot be evaluated based on bioburden data alone. For changes such as new materials or coatings, the impact from product-related inhibition and enhancement should be assessed along with the need for revalidation of the LAL method suitability. ISO 11737-3, Section 9.2.2 requires that reassessment of the impact of changes to the test method. 9.2.2, reassessment shall be performed for any change that can impact the test, for example, introduction of new materials, processing steps, product configuration changes, method of sterilization for post-sterilization testing, or a different product manufacturing site. This assessment shall include evaluation of the effect of the change on the outcome of determination. The results of this reassessment shall be recorded and suitability studies repeated as necessary. So what types of change might a manufacturer encounter? We could have a change in environment, which could be a change in manufacturing location or a change in cleanroom classification, perhaps. We could have a change in product design or materials, which could include change in vendors as well. Change in primary packaging, or a change, which could be a change in sterile barrier or internal packaging materials. We could also have a change in manufacturing, a change in manufacturing equipment, or the manufacturing process. Additionally, we could see a change in cleaning process, which could be a change in chemistries used in the cleaning process. A change in manufacturing location could affect the quantity and types of microorganisms on the product, which could mean the validated dose is no longer appropriate. The bioburden in the new environment could be more resistant to radiation. Additionally, the determined product family and representative product may no longer be appropriate. Let's look at some of the actions that might be performed in the situation where a change location occurs. This could be moving from one clean room to another in the same building, changing from one site to another, or a change of geographical location. This assumes no change in the device, raw materials, packaging, manufacturing process, or vendors. The change, again, should be documented in a formal change control. A risk assessment could also be useful to document the impact of the changes. Review of the classification of the cleanrooms, associated particulate and biological environmental data could be a useful exercise in understanding the impact to product associated with the change. Based on the evaluation of the impact, revalidation of the sterilization dose or confirmation through dose audit may be necessary. Microbial identification and an evaluation of resistance from testing may also provide insights on whether revalidation is needed. Given the potential for change in bioburden numbers and types, a review of product families and any representative devices is important to ensure the product families and the representatives are still appropriate. Additional considerations outside of irradiation validation might be the impact to aging, packaging, and distribution studies. If the storage or transportation condition of product is changed, for instance, in a different geographical location with hot, cold, or humidity extremes. Similarly, an assessment of existing product functionality studies should be evaluated for the impact of a change in potential temperature humidity extremes in comparison to historical studies. Also, assessing the impact of the change in location with regards to endotoxin levels where water is used in the manufacturing process, as the water source or water system may have different types and levels of microorganisms. Another change in the environment might be a change in cleanroom classification. For instance, a situation where classification has been downgraded to a lower level of filtration. This change would also have a potential to change the quantity or type of microorganisms present on the product, which in turn could affect the validated dose by it no longer being appropriate. Bioburden from the environment may be more resistant than previous microorganism types. The product family determined or the representative device may no longer be appropriate. A downgrade in cleanroom classification may warrant other considerations. This assumes no change in the device, raw materials, packaging, manufacturing process, or vendors. The change should be documented through a formal change control. A risk assessment, again, could be useful to document the impact of change. Review of classification of the cleanroom and associated particulate and biological environmental data could be useful in understanding the impact to product associated with the change. Based on the evaluation of the impact revalidation of the sterilization dose or confirmation through dose audit may be necessary. Microbial identification and any evaluation of resistance from testing may also provide insights on whether this revalidation is needed. A review of product families and any representative devices is important with any change to ensure the product families and their representatives are still appropriate. And additional considerations outside of irradiation validation may be the consideration on the impact to biocompatibility studies if there is a chance for increased particulate. A manufacturer may also consider a change in product design. This could be an addition of new components, an addition of coatings or agents, removal of components, or a change to product layout or product size. The possible product impact is the addition of new parts or materials may cause a change in the quantity or types of microorganisms present on the product and/or the validity of the test method validations. Possible impact to the test method is that it could affect bioburden recovery efficiency. It also could introduce or increase inhibitory substances requiring neutralization in the bioburden or sterility test methods. Inhibition may come from the presence of substances on or in the product, planned antimicrobial properties, or from substances that unintentionally cause inhibition. Possible impact to the radiation validation is that the calculated dose is no longer appropriate. Bioburden from the additional materials may be more resistant than previous microorganism types, and product families determined and representative products may no longer be appropriate. A change in product design may impact dose mapping or max dose studies, which include product functionality, product packaging, aging, and distribution testing. The change, again, should be documented in a formal change control. A risk assessment could be useful to document the impact of the change. Based on the evaluation of the impact, revalidation of the sterilization dose or confirmation through dose audit may be necessary. Microbial identification and an evaluation of resistance testing may also provide insights on whether revalidation is needed. A review of product families and representative devices is important with any change to ensure product families and their representatives are still appropriate. The applicability of existing test method validation should be considered from the change in design or materials and determination regarding whether revalidation of the methods are necessary. The impact of design or material changes to existing biocompatibility studies should be assessed to determine if new studies are necessary. Additionally, outside of irradiation validation considerations, the effects of the design or materials change could have impacted aging, functionality, packaging, distribution studies, and those should be assessed. Changes to the presentation of the product for irradiation need to be reviewed for impact dose mapping studies. A manufacturer may also have a change in raw materials, change in manufacturing material, but not a change in the product design. The potential impact to product is that the change in quantity or types of microorganism present on the product and the effect of the validity of the test method validations. Possible impact to test method validations is that it could impact the bioburden recovery efficiency. They may introduce or increase inhibitory substances requiring neutralization in the bioburden and sterility test methods, and they may impact endotoxin method validations if applicable to the product. Potential impact to radiation validation is that the calculated dose may no longer be appropriate. Bioburden from additional materials may be more resistant than previous microorganism types. The product family or determined representative device is no longer appropriate, and it may need to consider max dose studies for product functionality and aging studies. Consideration may also be needed to be given to the impact of the change to existing product functionality, biocompatibility, and endotoxin studies. Again, this process should be documented through formal change control. The completion of risk assessment for the proposed change could be useful, including a review of materials and technical specification documents, product bioburden data, and associated organism identification information. Completion of bioburden recovery efficiency validation, bioburden inhibitory substance screening tests, or BNF tests as deemed applicable. Revalidation of the sterilization dose if deemed applicable, or the performance of a dose audit study. Performing microbial identification on the bioburden plates to understand organism types and to determine if resistant organisms present and compare those with historical data. Assessing the appropriateness of product for inclusion in the product family or as a representative device. Refer to section 4 of ISO 11137-2. Additional consideration for changes in raw materials could be the consideration for the need to for the change to impact product functionality, aging, dose mapping, and biocompatibility studies, and if applicable, endotoxin studies. Manufacturer may also experience a change in vendor. Raw material, this would be where raw materials are the same specification, but they are coming from a different supplier. Possible product impact changes could be the change to bioburden types and quantities present on the product, which could impact the radiation validation in that the calculated dose may no longer be appropriate. Bioburden from the additional materials may be more resistant than previous microorganism types. Product families determined and representative products may no longer be appropriate. Documented action through a formal change control process is needed. Additionally, risk assessments can be useful for assessing the risk of the change, including the review of material technical specification documents, completion of bioburden testing for vendor change comparison, performing microbial identification on the bioburden plates to understand organisms present, revalidating the sterilization dose if deemed applicable, or performing dose audits, including bioburden testing and a dose verification study for a test for sterility. Assessing appropriateness of product for inclusion of a product family as detailed in section 4 of ISO 11137-2. And then additional considerations outside of the irradiation validation is to consider the impact to, if we're considering a product vendor change, considering the impact to functionality of that device as well as aging. If it's a packaging vendor, considering the packaging testing, distribution, and aging. And also to consider the impact to biocompatibility testing. Additionally, there may be a change in packaging. This could be a change in the sterile barrier tray or pouch or internal materials to maintain the configuration. For example, clips, ties, foam inserts, internal trays, backer boards, or information for use packets. Possible impact for internal packaging changes is a change to the quantity and type of microorganisms present on the product. This impact could affect whether or not the calculated dose is still appropriate. It could result in a change in bioburden types, which could be more resistant than previous organism types. It could also require a assessment of the product family and any representative devices. Additionally, it may require a need to consider dose mapping or max dose product functionality testing. The change to packaging documented in a formal change control, possibly containing a risk assessment to document the impact of the change. Based on the evaluation of the impact, revalidation of the sterilization dose or confirmation through dose audit may be necessary. Microbial identification, again, may be an evaluation of resistance from testing and may provide insights on whether revalidation is needed. A review of product families and any representative devices is important with any change to ensure product families and their representatives are still appropriate. The contribution of bioburden may be more likely from an internal packaging material change than a primary packaging change. Additional considerations outside of the irradiation validation is that there could be an impact of internal packaging to existing biocompatibility studies, and those should be assessed to determine if new studies are necessary. There could be a contribution of endotoxin from the change in bioburden coming from a change in internal packaging, and it may need to be evaluated. The effects of the packaging change to aging, functionality, packaging distribution studies should also be assessed. Changes to the presentation of the product for irradiation need to be reviewed for impact dose mapping studies. A change in manufacturing equipment or a change in manufacturing process could impact the change in quantity or types of microorganisms present on the product. This could impact the radiation validation and that the calculated dose may no longer be appropriate. The bioburden from the additional material may no longer be more resistant than previous microorganism types, and product families determined and any representative devices may no longer be appropriate. Assuming that there is no change in location, cleanroom classification, product design, vendors, raw materials, cleaning process, we would document the change through formal change control, complete a risk assessment for the proposed change, a documentation review, including review of the material technical specification documents. Given the large scope of this type of change, it is likely best to evaluate all of the listed items here. For example, a manufacturing change could be a leak test that could be performed in a water bath, or a change in type of sealer used in packaging, or a change in how adhesive is applied to a bond to components. All of these have different potential impacts. Bioburden has the potential to be affected by most changes which impact product families and their appropriateness for the sterilization dose and endotoxin levels. Additional considerations for manufacturing process and manufacturing equipment changes is to consider the impact to product functionality, aging, and if related to product packaging, consider the packaging and distribution testing. Consider the impact to biocompatibility testing. It is not likely that most changes would impact dose mapping, but this should be evaluated. The change to a cleaning process has the potential to change the quantity or types of microorganisms present on the product. It can also lend residues of the cleaning chemistry. This could impact organism recovery in the test method and require neutralization for either bioburden or sterility methods. Change in organisms could also impact endotoxin levels and existing biocompatibility studies. Changes to the cleaning process may impact radiation validation. For instance, the calculated dose may no longer be appropriate due to a change in the microbial reduction or efficacy of a cleaning process. The change in cleaning could contribute new bioburden, which could be more resistant organism strains. Additionally, the change may affect the product family designations and the selected representative device. Again, this should be documented through formal change control with potential completion of a risk assessment to provide supporting rationale, including review of materials such as technical specification documents, completion of bioburden testing for comparison due to a change in cleaning chemistry, performing microbial identification on bioburden plates to understand organisms present versus those found historically, complete bioburden recovery efficiency validation, bioburden inhibitory substance screening tests, or bacteriostasis fungistasis as deemed applicable, revalidation of the sterilization dose if deemed applicable, or performing a dose audit study, including bioburden testing and the sterility testing. Assessing the appropriateness of product for inclusion in a product family as representative device. And then those items outside of the irradiation validation could be an impact to product functionality and or aging. Consideration to biocompatibility testing, and also considering the impact of cleaning changes to endotoxins found on the device for those that devices that have endotoxin requirements. Thank you very much for joining us today.